question.
Okay, now we're
Did your daughters just say that?
So, we are talking about cancer, and I want to talk to you about the primary defense that we have against cancer. What we talked about last time was a multi-hit hypothesis, and we talked about 4 different hits. And these are changes to the DNA code, and the changes are in the part of the DNA that codes for proteins. So what this means is the amino acid sequence of the proteins, of some proteins, get changed. Does that make sense? So, the way our immune system works is our immune system, when a cell from the immune system is born, we'll call it a cytotoxic T lymphocyte, or a T killer cell. They don't use that word anymore. A CTL. When that cell goes out into the world through a technique or through a process I'm not going to talk about in detail here, you can take an immunology course and learn this stuff. This immune system cell knows the difference between shapes on proteins that are self and shapes on proteins that are not self. What I mean by self is, if you take your typical cell and that cell makes a protein and you were to look at, you've got some fold on the protein. If this is about 22 amino acids in size, that's got a shape, right? It's got this little 2-crescent kind of shape, right? So about 20 amino acids making it some particular shape. If this is a normal protein, if this is normal RB or whatever, that is made by my cell, or normal P53, that is a self shape, and my immune system will say, if my immune system bumps into that shape, it says, okay, you are me, you're okay, I'm not going to kill you. Very nice of the T cell, right? So if this is RB and it accumulates a mutation, better yet, this is one of the cyclins that accumulates a mutation so that it's stuck on and keeps driving the cell to go through the cell cycle. That mutation is going to be in some amino acid somewhere. So we could say, here's 20 amino acids, here's 20 amino acids. We could get, there's all kinds of, there's 1,000 different shapes on this thing, if you were to go on all different parts of it, right? So if there's a mutation in this, and that mutation is, maybe it puts an extra little wrinkle there with a positive charge, that is not a self shape anymore, right? Now when the T cell comes along, what the T cell does all day is goes, are you self? Yeah, you're self. Are you self? Yep, you're self. How about you? You're self. And if it bumps into this shape, it says, you are not self. And so it can recognize that a cell has non-self on it, and that T cell engages with a cell surface receptor on the cell that's got that defect in it. This cell surface receptor is called FAS. And the T cell has on it a cell surface receptor, which is the FAS ligand. When the FAS ligand engages FAS, the receptor gets turned on. What the receptor does is there is a protein called pro-caspase 8. Who knows about caspase 8? 1, 2? So pro-caspase 8 is some protein. And the active side of the protein is, say, this pocket right here. And that active side of the protein can't work because this stupid thing is in the way. But what FAS does is it cuts this protein right here, so that this part of it goes floating away, and we call that the pro domain. What we're left with is caspase, and it's active. Does anyone know what caspase 8 does? You guys didn't look that up? No big deal. Okay. The name comes from, I'm going to break it down for you. Where's a single letter code here? For cytosine, or if it's amino acid, it's cysteine. What's the 3-letter code? What is this?
Aspartate.
Aspartate. And then what does ACE mean? Cuts. Yeah, enzyme. So this thing was given this name because it is a cysteine caspase, cysteine aspartate cutting thing. What that means is this amino acid right here is a cysteine. And that what happens is another protein comes in, and if the protein binds in here, like this, if there's an aspartate right here, the cysteine will cut the protein like that. How many proteins, what percent of proteins have an aspartic acid, have an aspartate in their amino acid chain? A lot, like almost all of them. So this is a cysteine-based aspartate cutting thing. So they named it caspase. And this particular caspase actually has a very narrow range of things that it can cut. Okay? Very narrow range of proteins it can cut. Potentially it could cut a whole bunch. So once this thing is active in the prodomain, for the purposes of this class, the prodomain goes away and it does nothing else elsewhere. There's actually a protein which is named BID. And if BID gets processed this, BID loses its prodomain. And we don't call this pro-BID, but here we go. Does BID sound familiar to anyone? Who had BID? Right there, 2 people that did. What BID will do, so remember, if this T cell says, you have non-self on you, you have either a mutation or a virus in you. There's a virus in here, and the virus, all the shapes of the virus are non-self. So that's another way that a T cell would attack. So what this guy is saying is telling this cell is you need to commit suicide, and this is the process by which that message gets conveyed, in which this cell kills itself. So over here, there are 2 genes. Over here, there are 2 proteins. There's a protein called BAX and a protein called BCL2. And the job of BCL2 is to bind any BAX that's around. Oops. I said BAX because I was trying to write BCL2. Who had BAX? Anyone have BAX? One? Just one? Someone who skipped it today? Maybe it's Orlando or Katie. When you had BCL2, you're the only one with BCL2, I think. So normally, BAX is like a bad guy. It's going to help kill the cell. BCL2 is a protective factor, it keeps the cell alive. What happens is BID, once it has its prodomain off, BID comes along and prefers to bind here. Which means BAX is now out and available. Good thing we have a little bit extra BCL2. We'll sop that up. But then we'll process more BID. And now we have an extra BAX. And this BAX is deadly. This is a very long pathway. We're not anywhere near done here. Illustrated on the side. Max goes to the mitochondria and goes, boop, boop, boop. What do you think? Is that good for the mitochondria?
No.
The brains are kind of working for that thing.
Yeah. Does anyone know what's in this space here?
Protons.
Protons, yeah. From the electrochemical gradient that's formed for the proton pump that produces ATP, right? So, if you poke holes in this, the protons can leave, and you lose all the positive charges from here, which means the mitochondria can no longer generate ATP. If a cell is deprived of ATP, the cell is going to starve for energy and it will die. But in the electron transport chain, one of the proteins in that electron transport chain that was, if you took classes with me, the way I describe it is the electrons are kind of enticing a positive charge over them, and they say, "Psych, you can't have it," and off they go. Part of that electron transport chain, one of those proteins is called cytochrome C. Not only does Bax poke holes in the membrane, it takes cytochrome C out of the membrane and sticks it out here. And nobody had cytochrome C to clone. I've just not had luck with anyone PCR implanting that before. Not only is the proton gradient going to dissipate because there's holes in the membrane, but you can't continue the electron transport chain to rebuild these guys back up. The next thing that happens is cytochrome C is not normally out here in the cytoplasm. It's supposed to be in the membrane, in the inner membrane right here. It turns out there's a protein hanging out in the cytoplasm waiting for cytochrome C. And if it finds cytochrome C, it binds to cytochrome C, and that is the signal that turns this protein on. The protein is called APAF. There are all kinds of websites. You could say apoptotic pathway, and you can get this pathway that I'm drawing for you. You don't need to learn and memorize this. Isn't it sick that I know this off the top of my head? Anyway. Once cytochrome C complexes with APAF, and they form this dimer, APAF is actually this huge thing. It makes a new organelle in the cell. You've learned about the spliceosome, right? Now you need to learn about the apoptosisome. What this guy does is this guy takes caspase 3. Does anyone have caspase 3? What do you think it does to caspase 3?
Cuts off the pro.
Cuts off the prodomain. What do you think caspase 3 does? Huh? Same thing caspase 8 did, but even better.
So it's just bigger than everything.
Right. So see if I can come up with a description that would be similar to this. So the way I think of caspase is like a little samurai warrior with a little sharp blade, and it whoosh, cuts through things. Caspase 8 will, any feline that it sees, whoosh, gone to felines. I have cats, I love cats. Same thing, I like cats. So it's specific for one kind of thing. Caspase 3 is like the most incredible ninja you've ever seen, holding a sword between each finger and whipping them all around like this. Anything it bumps into, it chops up and chews up into bits. Any protein it bumps into, toast. So, the things that make the cell hold together and hold its shape, there's like girder networks inside the cell that hold the cell in its shape. All those proteins get chopped up, which means the cell doesn't have structural support. It just kind of like gets watery and round and blubby, and the membrane starts falling off in little. If you have a big, huge bubble like this, let's say I have a big, huge bubble of water like this, and I'm in outer space, it's sitting here like this. Over time, that bubble is going to pick up kinetic energy. It's going to be flopping around, and you can imagine it would kind of dissipate into smaller bubbles of water, right? When the internal parts of the cell get blended by caspase 3, the cell shrivels up, and we saw that in that picture last time. That rounded up cell that's got all those round little blubby things. The German word for it, I can't remember, but it basically means cauliflower cell. The cell has a cauliflower kind of look to it. Something caspase 3 does is it takes pre-DFF, converts it into active DFF. Active DFF goes into the nucleus. Can you see where this is going? Can anyone guess a name of this? DFF.
Death. Something, something.
What?
Death, something.
Death, something, something. That's a good guess, no? What do you think this stuff is? DNA. DNA fragment factor. It's the DNA fragmentation factor. And it was identified because if some biochemists purified this protein and put it into a thing that had DNA, and the DNA got all fragmented into little bits. So they creatively named the protein DNA fragmenting factor. We're biologists. The DFF comes in and cuts the DNA up into tiny little bits. Chops the genome up. So the cell cannot generate energy because the proton gradient is gone. It can't regenerate the proton gradient because we screwed up electron transport chain. And it can't express any genes. What this guy has effectively done is said, you are not self. There is something not self about you. You are not me. Therefore, you need to commit suicide. I'm going to activate I'm going to activate the ninjas inside of you. And you will chop yourself into little bits, and then you're gone. This is our primary defense against cancer. Mutations that happen in the genes, if a cell has accumulated a lot of mutations, it will have a whole bunch of not self that it shows to the outside world. And so your T cells are the critical component of that. There is this disease called HIV, AIDS, which is caused by HIV. And HIV infects T cells and kills them. So that people don't have this defense against non-self. People who get infected with that sometimes get these lesions called Kaposi sarcoma lesions. Have you heard of this? They're basically like a precancerous thing. It's caused by herpes virus, actually. Because the body can no longer recognize self, or can recognize these infectious stuffs. Okay, this is supposed to be an X, isn't it? So, this pathway that I've just outlined for you, the generic term is programmed cell death. When it was first discovered, it was given this name. And I think it was discovered in about the 70s. Back when I was just a wee little lad, right? And you were just a twinkle, no, right? So someone who has never seen this word before. Who wants to pronounce this word for me?
Apoptosis.
Apoptosis. No. Try again.
Apoptosis.
Apoptosis.
Apoptosis. I should have spelled it that way then.
The P is silent, of course. And then in Greek, the P is silent. Apoptosis. repeat a cycle. If you go out in the research world and someone calls it apoptosis, that person has not been studying apoptosis very long. They don't know how to pronounce it. Or they were taught by someone who pronounced it incorrectly. Apoptosis. Say it, everybody. Apoptosis. Great, thank you. I would be mortified if you went out into the world and started saying to people, I learned about apoptosis today in class. Okay, so please don't do it. So programmed cell death is kind of the generic term for how a cell committing suicide and destroying itself, chewing itself up into bits from the inside and dying. This is a pathway of killing itself. So this is a pathway of programmed cell death. They named this pathway apoptosis. There is another pathway in which lysozymes get activated, and lysozymes will eat up the internal components of the cell until the cell eventually has nothing in it and it basically falls apart. That is a different way that a cell can commit suicide, and that pathway is called, oh boy, I can't believe I'm forgetting this. Autophagic degeneration. Parts of the cell's eating itself alive. That's basically what it means. So there are multiple ways that a cell can commit suicide, can commit programmed cell death. Apoptosis is the very well understood one. Autophagic degeneration, people are like, I don't know. They don't know the genes that control it very much. There is another way that a cell can commit suicide, and I find this amusing every time I teach this. Who had a gene called BAD? You got the BAD gene? You're the only one with the BAD gene? What was that? Submarine coming in? Okay, there's this gene called BAD. And BAD is phosphorylated and inactive in the cell. And the reason is BAD gets made, but there is protein kinase B, which some people have decided to call AKT, for whatever reason. It's a protein kinase, and anytime it finds BAD, it phosphates on it, says, time out. It's hockey playoffs now. You guys watching the hockey playoffs? Well, what's the timeout when they get a penalty? They're in the penalty box, right? BAD goes into the penalty box forever. It gets phosphorylated and stuck in there. Oh, every now and then a phosphate will fall off, and it'll get its foot out the door, and the ref will come by and say, I'm going to give you another penalty. Back you go. Right? Phosphate goes back on. And BAD spends its whole existence in the phosphorylation box. Okay? How is this protein kinase B, or AKT, how is that regulated? Turns out, cells are have a low confidence self-esteem, low confidence level. Cells are like my 4- and 5-year-olds in that they need continual reassurance. Oh, that was very good. You're doing a great job. Hey, way to put those shoes on. Did you comb your hair? I bet you can do a great job of that. Cells need constant stimulation from the outside to say, good cell, way to go, good job. And they have, these things are called growth factors. And these are chemicals that are released. And in a healthy person, these things are released. And they go running around, float around through the body. And if they bump into a cell, they're the little signal that says, good cell, way to go, nice cell. Give me a little scritch. When the growth factor binds to its receptor on the surface of the cell, it sends a signal in that turns these guys on. If this signal should go away, this signal will stop, these guys will go away. And BAD will eventually shift back onto the ice, and it's playing the game. What bad does is it goes over here, and it prefers to interact with BCl2. And it'll knock backs off of BCl2. Whereby BCl2 pokes holes in the membrane, causing the protons to leave. No more ATP gets made. It knocks cytochrome C out, so you can't recreate the proton gradient. Cytochrome C turns on the super ninja, which chews the cell into bits, and the cell dies. So the reason I think this is such an interesting pathway to put up here is, if you took a cell out of isolation, and you put it in a tube, the default existence for that cell is to commit suicide. Isn't that funny? Every cell in your body has this pathway of apoptosis in it, primed, ready to go. Every cell in your body is, at a drop of a hat, ready to commit suicide. And if the growth factors stopped being produced in your body, your cells would all die by this process. Is that interesting? The default existence of your cell is suicide. I think it suggests eat healthy, so that your cells are healthy. Girder network is broken. Stem goes floating over here and activates BAX through this process. So Bach is going to regulate something inside the cell like this. Yeah.
Well, this is like a change of subject, but like, what does it have to do with cancer?
What does this have to do with cancer? If a cell accumulates mutations in cell cycle genes, or gene like P53, that P53 gene, that, remember, guardian of the genome, right? If P53 has a mutation in it, that mutation can be detected by this T cell over here. And the T cell says, you have accumulated mutations. You are not self anymore. You're not the self that should be existing in here. There's a problem with you, and so I'm going to tell you to commit suicide.
But how come this doesn't always work?
So this is the fifth hit in the multi-hit hypothesis. The fifth hit, thank you for that, the fifth hit in the multi-hit hypothesis is you need to screw this pathway up. So if I get a mutation such that the ninja, we knock out this gene, that no ninja comes to exist, is the T cell still going to be able to cause the cell to commit suicide?
Well, it's not going to be happy, but not as dramatic.
It's not as dramatic.
It's probably still going to die, but it might stay.
Why would it die?
Because
There's no energy being, yeah, the mitochondria are dead. A cell that can't generate ATP is going to be a dead cell. So where would there have to be a hit? If you were to hit a gene in this pathway, where would the hit be to shut down all programmed cell death? BAX. If you can knock out BAX, like this, if a cell knocks out BAX, it doesn't matter if big gets activated or bad gets activated, you're never going to release BAX to screw up the mitochondria. What's the other possibility? Caspase 8, no, because you could activate this way.
Screw the T cell.
You could screw up the T cells. Yeah.
Path one.
Huh? How else could you? One last one. You could take something away, or you could add something. BCl2. If you got chromosomes to cross over incorrectly, such that a really, really strong promoter was upstream of BCl2, you would make so much BCl2 that if you that it would soak up all the BID, it would soak up all the BAD, it would soak up all the BAC, it would soak up all those guys, and still be able to hold on to the BACs. BACs would never get released to disrupt this. BCL2 stands for B cell lymphoma. And it turns out this is the #1 way. If a cell has become cancerous to the point that it's formed a tumor, my bet is it has found a way that is making massive amounts of BCL2. So that all those insults come in and they only get as far, and the cell is fine the rest of the way down. Yeah, you could knock out FAS. That would help do it. Okay, this pathway of apoptosis is more important for more than just cancer. They've done interesting things. So someone went in to the mouse genome and they knocked out BAX. So the mice can't produce BAX, and the mice cannot, the cells cannot commit suicide by this process. And they took a BAX knockout mouse and a BAX knockout mouse, they bred them together, and they had a litter, and the mice had enormous heads. And they had, in between their fingers, there were skin webbing. Turns out this pathway of committing suicide, there exist at various times cells in our body which are used to help the other cells get set up properly, and once everything's all set up properly, these cells are like, oh, no, I'm in the way, and they commit suicide to get out of the way so everything can happen normally. So when mammals are in utero, developing, and the limbs are just these little stubs, there are cells that, in lines, they somehow figure out where they are in relation to this side and this side. They're communicating back and forth this way. And cells in lines here commit suicide such that the paddle becomes separated into individual digits. Who's this big time swimmer at the Olympics?
Michael Phelps.
Michael Phelps. I think he has a little webbing here in his fingers. First of all, he's got enormous hands, but he has a little bit of webbing there. I see that, and I think he's got a problem somewhere. He's got a mutation somewhere in his apoptotic pathway. And I'm going to say maybe he's going to get cancer, or more likely to get cancer possibly than someone else. Why were the heads of the mice enormous? And this is where we learned this from the study. Neurons are made, and they have a bunch of helper cells hanging around saying, you need to grow your axon this way, grow your axon this way, because you're supposed to connect somewhere over here. And there's a whole bunch of cells guiding during development when neurons are forming. Getting the neurons, there'll be neurons in specific areas that send their axon to other specific areas. And they know to send it because it sends its axon along a chain of cells. Once the axon gets to its destination, all that chain of cells, most of those can go away now because the hard wiring of the brain is set in place. So most of those cells commit suicide. What happened in the mice is those cells couldn't commit suicide through this apoptosis process. So those cells persisted. And you say, oh, the mice have huge brains, they must have been really smart. They had huge brains because it was filled with not neurons, and the mice were actually not very smart. Like if you have a normal mouse, it's that smart. These guys were that smart. I don't know how you test that. What's your favorite color? Blue, no green. There's another pathway that comes in and feeds in here. And if a cell has double-stranded RNA, viruses have double-stranded RNA genomes. And we talked about this panic response when we were talking about the microRNA. And so they tested for that in that paper. If a cell detects double-stranded RNA, this thing called PKR comes in, and PKR activates, it comes in through a different BCL2 interacting protein and initiates this pathway this way. So it's interesting. Viruses, let's see, which virus was it? Poliovirus. Poliovirus makes a protein that binds onto caspase-3 and prevents it from doing its work. So that all this stuff persists and hangs around. The cell doesn't get all chopped up into bits. A number of papillomavirus causes warts. Papillomavirus strain 16 and strain 18 they infect cervical cells in females. And that virus has a gene called E2, no, not E2F.
It's
It
2
binds-
and 6.
Huh?
E2 and E6.
E2 and E6 is it in E7 is not. It's E6 that does it. I think.
Yeah, I think one of them binds to RB and one binds to P53.
Yeah. They shut down also this somehow. How did they do that? I'm trying to remember. E2. E2F is named for the E2 protein, right? Does that work? Anyway, those viruses make a protein that can interact with these guys at this level. Protective viruses cell up there. Viruses are trying to make an infected cell last longer. So they disrupt this process so that the cell is forced to stay alive. Okay. Do you guys have questions about this stuff?
You said we don't have to memorize all of that, right?
Good God, no. If you go in and type in apoptosis pathway on Google Images, there's 5,000 images that illustrate this thing right here. What other genes did I not talk about that you guys had?
Back.
Back? Back and BOC interact with this area. They're called BCL2 family members, all of these things. BIM is another one. A9 or something like that, I don't remember, A, something like that. There's a bunch of different genes. They're called BCL2 family members because they interact with BCL2. BCL2 regulates whether they're allowed to control mitochondria or not. Okay, that's it. Thank you for a fun semester. Evaluations are on Blackboard. Yes? So fill those out. And take home final exam. Get that out to you as soon as I can. Do a week from tomorrow. Yes? That's the last day of finals? Those of you who are graduating, have a good time.
If you pass the class. I just said if they pass the class.
Oh, yeah. Oh, yeah.
This is probably not necessarily graduation.